You all know what DNA is, right? Double helix, CATG pairs. Yeah, of course you do, because you're not an amoeba. How about DNA replication? Know that one, wise guy? No? Then buckle up, kiddo, because I'm contractually obligated to tell you about it. It all starts with this little guy, a helicase enzyme. He takes the DNA and unzips it, just like a jacket, taking the two strands and pulling them apart. Zip. Just like that. So now we got two half strands of DNA in what's called a replication fork. This is the part where the next enzyme, primase, comes along, and using RNA makes a little piece called a primer. See how that works? Primase, primer, see? This primer is then used by a whole other enzyme, DNA polymerase, to start attaching DNA bases to the 5' end, working towards the 3' end. This is the only way it works, like a one-way street. This only happens on one strand, the leading strand. The lagging strand has to keep up by using smaller sections called origami frag... No, that's not right. Okazaki fragments, that's it. Okazaki fragments help fill the lagging change by being put on one little section at a time. Primer, Okazaki. Primer, Okazaki. You get the picture. But these little primers can't stay there. That would be like leaving the bones in an ice cream cone before you serve it. Just unnatural. So this fella, exonuclease, comes along and picks out all the bones. All the primers. So that way DNA polymerase can come and put a fragment there, filling in the gaps. Lastly, the enzyme DNA ligase comes along and seals up the two new strands of DNA so they're ready to be sent off. This process is defined as semi-conservative. And no, not conservative like your uncle's Facebook posts. Conservative here means that these new whole DNA chains contain one old conserved strand and one new one. Okay. Like the 6th take. Have fun editing this, Tessie. Next, I'm going to answer one complex question. How does a cell know that it's supposed to code for a human, or a dog, or a lizard? To put it simply, it's the process of transcription and translation that reads the genetic code properly. It takes some serious know-how to read the information on a chromosome. They're long and complex strands that don't contain a lot of important information, but the little bits that do matter are called genes. When a gene is expressed, that results in the creation of a protein. The first thing that happens is transcription, which is how DNA creates an mRNA strand with the help of a DNA polymerase that binds to a promoter at the start of a gene and pries it open like a bag of potato chips, creating two strands, the template strand and the non-template strand. The template strand, as the name suggests, is what the polymerase uses to make the mRNA. The process is fairly similar to DNA replication, except this time we're creating one mRNA strand and not two DNA strands. Once the mRNA is constructed, the DNA is closed back up and the mRNA leaves the nucleus where all the genetic material is stored. It finds its way to a ribosome. This is where the translation comes in. The ribosome reads the mRNA like a computer would read a floppy disk, and then produces a certain protein by pairing the codons to anticodons. The tRNA that the ribosomes use to pair up with the codons has amino acids that correlate with a specific codon chain. The ribosome uses this information like a blueprint on a factory floor to link several amino acids together and form a polypeptide chain, which then heads off into the cell to be used for other functions. To sum it all up, transcription is how the cell takes a gene and creates an mRNA strand with it, and translation is how a ribosome uses that mRNA and tRNA to make a polypeptide chain, and ultimately a protein.